Students, today I bring for you a new chapter, that is cell cycle and cell division. So previously we just discussed about biomolecules, and now we are going to cover this chapter. In this chapter we are going to cover 5 major important topics which are in your syllabus, that are cell cycle, M phase, significance of mitosis, meiosis and significance of meiosis. So first of all, in your NCERT it is written that growth and reproduction are characteristics of cells, indeed of all living organisms. Here you can see growth and reproduction of all organisms depend on division and enlargement of cells. The mechanism of division and multiplication of cells together constitutes the cell reproduction. Next is the cell cycle, and cell cycle means cell division is a very important process in all living organisms, and during the division of the cell, DNA replicates and cell growth also takes place. All the processes that are cell division, DNA replication and cell growth, they occur in a proper coordinated way to ensure the proper division of the cell and the formation of progeny cell containing the intact genome. So here cell cycle means it is the life period of a cell during which a cell synthesizes DNA, growth and divides into two daughter cells. Cell growth, that is also called cytoplasmic increase, is a continuous process. DNA synthesis occurs only at a specific stage. So according to proper defining cell cycle in your NCERT, it is given that now the sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell and divides into two daughter cells is termed as cell cycle. And cell growth means we can say cytoplasmic increase, it is also a continuous process. Next in cell cycle, now duration of cell cycle varies in each organism and each cell type. So the cell cycle duration is all different from every organism and the cell type also. Duration of a typical eukaryotic cell cycle, example human cell, is about 24 hours, and in yeast it only about 90 minutes. Okay, so it vary from organism to organism and also from cell type to cell type. Next, it is given it has two basic phases. Cell cycle divides into two basic phases, interphase and second is the M phase, or we can say mitosis phase. First of all, interphase or resting phase. Interphase is the phase between two successive mitosis phases. It is the phase between two successive mitosis phases. Okay, you can see in this diagram, it includes cell growth and DNA synthesis. It lasts more than 95% of the duration of the cell cycle. So here it is said it lasts more than 95% of the duration of cell cycle. In your NCERT, it is given that in the 24 hours average duration of a cell cycle, a human cell division proper lasts for only about 1 hour. The interphase lasts more than 95% of the duration of cell cycle. Okay, and interphase has three phases. Okay, G1 phase or gap 1 or antiphase also called, synthesis phase, S phase and G2 phase or gap 2 phase. In your NCERT, two more terms are given, that are karyokinesis and cytokinesis. Karyokinesis means separation of daughter chromosome and cytokinesis means division of cytoplasm. Okay, so interphase also called resting phase and divided into three phases: G1 phase, S phase and G2 phase. Okay, first of all, G1 phase or gap 1 phase or antiphase. What does it mean? The first point, it is said it is the first growth phase. It is the first growth phase It is the interval between mitosis and DNA replication. Interval of the between the mitosis and DNA replication, and main events of the G1 phases are continuous growth of the cell becomes metabolically active, prepare machinery for DNA replication, and synthesizes RNA and protein. Okay, in your NCERT it is given it is cell is metabolically active, continuously grow but does not replicate its DNA. Okay, does not replicate because replication occur during S phase or synthesis phase. Here coming to the second phase, synthetic phase. In this DNA replication takes place, amount of DNA per cell doubles but chromosome number is not increased. Means your NCERT is saying that if the initial amount of DNA is denoted as 2C, then it increases to 4C, 4 chromosome. However, there is no increase in the chromosome number. Okay, so if the cell has a diploid or 2n number of chromosomes at G1 phase, even after S phase, the number of chromosomes remain the same, that is 2n. In animal cell, replication begins in the nucleus and the centriole duplicates in the cytoplasm. You can see in this diagram. Next is your next phase, that is G2 phase. G2 phase or gap 2 phase is the second growth phase. Cell growth continues, synthesis of RNA and proteins continues, and cell is prepared for mitosis. Okay, it occurs in the G2 phases. Now the important or main phase, that is called M phase or mitosis phase. It represents the actual cell division, which is called mitosis. In human cell cycle, it lasts for only about 1 hour. Okay. M phase includes karyokinesis, that means nuclear division, and cytokinesis, that means division of cytoplasm. You can see the diagram. There are two different type of karyokinesis, cytokinesis. Karyokinesis, that is nuclear division, is divided into 4 phases: prophase, metaphase, anaphase, and telophase. And cytokinesis is the division of cytoplasm. Next, some points about M phase. Some cells do not show division. Example, heart cell. Here it is said, some cells do not show division, that is heart cell. Many other cells divides only occasionally to replace damaged or dead cells. Okay. In your NCERT it is also said that some cells in the adult animal do not appear to exhibit division. Example, heart cell. Okay. And many other cells divide only occasionally as needed to replace cells that have been lost because of injury or cell death. These cells do not divide further and they exit G1 phase to enter an inactive stage, that is called quiescent stage or G0 stage of the cell cycle. In the G0 phase, the cells remaining metabolically active but no longer proliferate unless they called on to do so, depending on the requirement of the organism. Here the same points are given. You can see the cells that do not divide further exit G1 phase and enters an inactive stage called quiescent stage. They are metabolically active but do not proliferate. Okay, you can see G0 phase is also mentioned in this diagram. Next is your mitosis. Before starting mitosis or M phase, it is also said in your NCERT that mitotic cell division is only seen in the diploid somatic cells. However, there are few exceptions to this where haploid cells divide by mitosis. For example, male honeybee, and against this the plant can show mitotic divisions in both haploid and diploid cells. Okay, and also it is also given about the onion root tip cell, mitosis in onion root tip cell, which it is also mentioned. It has 16 chromosomes in each cell. Okay, and some questions are also in your NCERT. So let's start. Mitosis. It is a cell division occurring in the somatic cells. It is also called equational division. It is also called equational division as the number of chromosomes in the parent and progeny cells are the same. Mite is generally seen in the diploid cells. Okay, it also occurs in haploid cells of some lower plants and some social insect. Example, honeybee, male honeybee. It involves major reorganization of all cell components. Here you can see the karyokinesis of mitosis has 4 stages: prophase, metaphase, anaphase and telophase. You can see in this diagrams, it is the diagram on the that is taken on the proper electron microscope, and this one is the diagram. It also I think in your NCERT, and as well as you can find this diagram in other different internet sites. Okay, so let's start the first phase, that is called prophase. So mitosis is called equational division. Remember the point. Prophase. It is the longest phase in mitosis. The first point it is said it is the longest phase in mitosis. It follows the S and the G2 phase of interphases. In the S and G2 phases, DNA molecules are intertwined. So here the diagram, it is also in your NCERT. So some more points, characteristics, event. But before starting this, coming to our NCERT, it is said that S and G2 phases, the new DNA molecules formed are not distinct but intertwined, means mixed. Okay, and prophase is marked by the initiation of condensation of chromosomal material. The centrosome, which had undergone duplication during S phase of interphase, now begins to move towards the opposite pole of the cell. So the characteristics events of the prophase is the chromosomal material, that is chromatin fiber, are untangled and condensed to form mitotic chromosomes. They are seen to be composed of 2 chromatids attached together at the centromere. Centrosomes begins to move towards opposite pole of the cell, and each centrosome radiates out microtubules called asters. The 2 asters together with spindle fiber forms mitotic apparatus. Asters can be seen in other diagrams. We can see that asters. Okay, so this is the main 2 characteristics event of the prophase. Cells at the end, it is also said, listen, in your NCERT also, cells at the end of the prophase do not show Golgi complexes, endoplasmic reticulum, nucleolus and the nuclear envelope. So at the end of the prophase, or we can say late prophase, cells do not have Golgi complexes, ER, nucleolus and nuclear envelope. Remember this point. Next is metaphase. In metaphase, the nuclear envelope completely disintegrates, as you can see in this diagram. Hence, the chromosomes spread through the cytoplasm of the cell. Chromosomes condensation is completed. They can be studied easily under the microscope. They will have the 2 sister chromatids. As I told you, what are asters? Now, these are the asters in this diagram. You can see these are the asters. Okay, they have chromatin fibers attached. Next, chromosomes comes to lie at the equator. The plane of alignment of chromosomes at metaphase is called metaphasic plate. As you can see, chromosomes are lie at the equator. Okay, this is called metaphasic plate. Okay, this plate is called metaphasic plate, proper equator in the middle. The spindle fibers from both poles are connected to chromatid by their kinetochores in the centromere. It is also said the spindle fibers, you can say labadia, from both the poles are connected to the chromatid by their kinetochores in the centromere. Okay, so what are kinetochores? Now these are the kinetochores. The whiting portion you can see in this diagram, this is called kinetochores. Okay, and the middle portion is called centromere. Next phase is your anaphase. What is anaphase? Now it is the shortest phase in the mitosis. Centromere of each chromosome divides longitudinally, resulting in the formation of 2 daughter chromatids or chromosomes with the future daughter nuclei. As the spindle fiber contract, the chromatids move from the equator to the opposite poles. As you can see, like this say, they become, and here also some points are about anaphase, and that is the arm of the chromosome trailing behind. As you can see this diagram, the arm of the chromosomes trailing behind. Trailing means they are attracting by the spindle fibers, okay, and trailing like this, okay. Next is your telophase. What is telophase seed? Now telophase means chromosomes cluster at opposite pole and uncoil into chromatin fibers. So chromosomes are uncoiled, or you can say decondense, and lose their individuality. Chromosomes decondense, okay, NCERT line, decondense and lose their individuality. Nuclear envelope assembled around the chromatin fiber, thus two daughter nuclei are formed. Nucleolus, Golgi complex and ERR reappear. In which stage? Now in telophagic stage. Nucleolus, Golgi complex, ERR reappear. The spindle fibers disappear, and the spindle fibers disappear in the telophage. Next is your cytokinesis. Cytokinesis means division of cytoplasm. So it is the division of cytoplasm resulting in the formation of two daughter cells. It starts when telophage is in progress. Next, cytokinesis animal cell. You can see in animal cell a cleavage furrow is appeared in the plasma membrane. Cleavage furrow, this portion, okay, cleavage furrow, okay. It appears in the plasma membrane. It gradually deepens from the both sides. It deepens, okay. It deepens from the sides like this, and joins in the center, dividing the cytoplasm into two portions. Cleavage furrow, remember the term, okay, cleavage furrow. But in plant cell, there is no cleavage furrow is formed. Instead of cleavage furrow, plant cells develop cell plate. Okay, plant cells develops cell plate. It is different from the cytokinesis in animal cell due to presence of cell wall. In plant cells, the vesicles formed from Golgi bodies accumulated at the equator. So, from where the plant cells, where the cell plate is formed, in plant cell, vesicle from Golgi body, Golgi complex accumulates at the equator. Okay, it grows outward and meet the lateral wall. They fuse together to form cell plate like this. Growing cell plates, you can see, and these cell plates are made by vesicles from the Golgi bodies. It separates the two daughter cells, and later cell plate becomes middle lamella. So like this, it separates the two daughter cells and it forms the middle lamella. Okay, clear? It is occur in the plant cell. Okay, animal cell, cleavage furrow is formed. Next, in your NCERT, there is some term about that is given that in some organisms karyokinesis is not followed by cytokinesis, as a result of which multinucleate condition arises leading to the formation of syncytium. Okay, the term syncytium, that is which is the example of liquid endosperm in coconut. Remember the term syncytium. Okay, it is also given here. During cytokinesis, organelles like mitochondria and plastids get distributed between the daughter cells, and in some organisms, karyokinesis is not followed by cytokinesis. As a result, multinucleate condition syncytium arises. That is the liquid endosperm in coconut. Here are the overall steps. You can see interphage, how the nucleus divided, centrosome is also present, prophage, prometaphage, metaphage, and then anaphage, telophage, cytokinesis. It is the overall steps of the mitosis. Now coming to the significance of mitosis. Coming to significance, Now first point is given, it produces diploid daughter cells with identical genome. It helps to retain the same chromosome number in all somatic cells. It helps in the body growth of multicellular organisms. Mitosis in the meristematic tissue helps in a continuous growth of plants throughout the life. It restores the nucleus-cytoplasmic ratio that disturbed due to cell growth. Nucleus-cytoplasmic ratio. It helps in cell repair and replacement. Example, cells of upper layer of epidermis lining up the gut and blood cells. In your NCERT, the same things are given. Reproduction of diploid daughter cells and identical genetic complements. The growth of multicellular organism is due to mitosis and a disturbing ratio of the nucleus and cytoplasm. So therefore, it is essential for the cell to divide to restore the nucleus-cytoplasmic ratio. Okay, and the main contribution of mitosis is the cell repair. Next is your meiosis. Meiosis is the division of diploid germ cells that reduces the chromosome number by half, forming haploid daughter cells or gametes. So simply, meiosis, it ensures the production of haploid phages in the life cycle of sexually reproducing organisms, whereas fertilization restores the diploid phages. It occurs during gametogenesis. Okay. So the key features that are mentioned in the NCERT also, NET involves 2 cycles, meiosis 1 and meiosis 2, but only a single cycle of DNA replication. It involves pairing of homologous chromosomes and recombination between them. Meiosis 1 begins after replication of parental chromosome to form identical sister chromatids at the synthetic phase. 4 haploid cells are formed at the end of meiosis 2. So these are the characteristics or key features of the meiosis. So it is grouped under 2 different portions. Meiosis 1, which divides into prophase 1, metaphase 1, anaphase 1, telophase 1, and meiosis 2 includes 4 stages: prophase 2, metaphase 2, anaphase 2, telophase 2. First of all, meiosis 1, prophase 1. It is a typically longer and more complex. Meiosis 1, prophase 1 is the more complex and longer phase. It includes 5 phases based on chromosomal behavior that are leptotin, zygotin, pachytin, diplotin, and diakinesis. These are the 5 stages of the prophase 1. That's why they are longer and more complex. First of all, leptotin or leptonema. In your NCERT, it is said that leptotin stage, the chromosomes becomes gradually visible under light microscope. The compaction of chromosomes continues throughout the leptotin. So the main point of the leptotin is the chromatin fibers become long slender chromosomes and nucleus enlarges. Okay, compaction of chromosome. As you can see, nuclear envelope and the compaction of chromosomes occur. Second, zygotin or zygonema. And what does it say? Now, chromosomes becomes more condensed. Chromosomes becomes more condensed, and similar chromosomes starts pairing together. That is called synapses. The term also given in your NCERT. You can see in zygotin stage, chromosomes start pairing together, and this process of association is called synapses. Such paired chromosomes are called homologous chromosomes. And here you can see the help of a complex structure called synaptonemal complex. So how the synapses occur? The pairing of similar chromosomes occur by the synaptonemal complex. Okay. The paired chromosomes are called homologous chromosomes. Each pair of homologous chromosomes is called a bivalent or a tetrad. Well, the tetrad word, that is in your NCERT. So here you can see synaptonemal complex forming like this. Similar chromosomes starts pairing together. The process is called synapsis, and by a complex structure that is called synaptonemal complex. Next is pachytin or pachynyma. comparatively longer phase. Bivalent chromosomes split into similar chromatids. This stage is called tetrad. So here particularly the tetrad word, that is for Pachitin stage, but in your NCERT it also given in the side of the bivalent word. Bivalent chromosomes split into similar chromatids. This stage is called tetrads. Appearance of recombination nodule at which crossing over occur. Crossing over means you can see here. Crossing over is occurred here like this. Crossing over. Okay. Crossing over means the exchange of genetic materials between non-sister chromatids of a two homologous chromosomes, and it occurs in presence of enzyme that is called recombinase. Okay. Remember the crossing over word and the enzymes also. Recombination is complete by the end of Pachitin. In your NCERT, it is also same points given that 4 chromatids of each bivalent chromosomes becomes distinct and clearly appear as tetrads. This stage is characterized by the appearance of recombinant nodule and the site at which crossing over occurs between two non-sister chromatids of the homologous chromosomes. Crossing over is the exchange of genetic material between the two homologous chromosomes, and it is an enzyme mediated process, and the enzyme involved is the recombinase. Okay, remember the term crossing over and the enzyme recombinase. Next, diplotin or diplonema. Dissolution of synaptonymal complex occur. Dissolution of synaptonymal complex occur. The recombined homologous chromosomes of the bivalent separates from each other except at the site of the crossing over, and the X-sept structure. The X-sept structure like this structure is called chiasma or chiasmata. Okay, this structure is called chiasmata. It's an X-sept structure. In oocytes of some vertebrates, diplotin lasts for months or years. In oocytes of some vertebrates, the diplotin lasts for months or years. Next is your diakinesis. The final stage of meiotic prophase 1 is the diakinesis, and this is marked by the terminalization of chiasmata. Terminalization of chiasmata, chromosomes are fully condensed. The meiotic spindle fiber originates from the pole to prepare the homologous chromosomes for separation. Nucleolus and nuclear envelope disappear. Remember, nucleolus and nuclear envelopes are disappear. Okay. Next, meiosis 1, metaphase 1. These are the prophase and 5 different stages. Next is your metaphase 1 of the meiosis 1. Spindle formation is completed, as you can see in this diagram, where the prophase in metaphase, the differences you can see. The chromosomes align on the equatorial plate, means in the central plate, equatorial plate. The microtubules from the spindle attach to the paired homologous chromosomes. The microtubules attach to the pair of homologous chromosomes. Next is your anaphase. Anaphase is the homologous chromosomes separate while sister chromatids remain associated at their centromeres. It is the anaphase. You can see metaphase, anaphase. Okay. Next, telophase 1. The nuclear membrane and nucleolus reappear. Remember, the nuclear membrane and nucleolus reappear, and two haploid daughter nuclei are formed. Two haploid daughter nuclei are formed. This is called a dyad. What is it called? It is called a dyad of cell. Okay. Next, after this, cytokinesis may or may not occur. After this, cytokinesis may or may not occur. After a short interphase, it is followed by meiosis 2. This short stage between two meiotic division is called intricacies. Okay. In intricacies, there is no replication of DNA occur. Okay. Next is your meiosis second. Meiosis second, it resembled the mitosis. It has 4 phases: prophase 2, metaphase 2, anaphase 2, telophase 2. Okay. So first of all, prophase 2. It is initiated immediately after cytokinesis. The chromosomes again become compact. Nucleolus, nuclear membrane disappear in both nuclei. Okay. Metaphase 2, here you can see the chromosomes align at the equator and the microtubules from opposite poles of the spindle gets attached to the kinetochore of the sister chromatids. Anaphase 2, here you can see it begins with the simultaneous splitting of the centromere of each chromosome, which holding the sister chromatid together, thus they move towards the opposite pole of the cell. It is the anaphase 2. And the last one is the telophase 2. Telophase 2 of the meiosis 2, it says the two group of chromosomes once again gets enclosed by a nuclear envelope. Cytokinesis follows, resulting in the formation of tetrad of cell, that is 4 haploid daughter cells are formed. Okay. And the overall steps of the meiosis, overall steps, prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. Here in meiosis 1, meiosis 2, it also said parent cell, interphase, prophase 1, metaphase 1, anaphase 1, telophase 1, and 2 daughter cells are formed. In meiosis 2, you can see prophase 2, metaphase 2, anaphase 2, telophase 2, and 4 haploid cells are formed. Now the last topic, significance of meiosis. And it is said that it conserves the chromosome number of each species. Main point, it reduces the chromosome number by half and increase the genetic variability in the population of organism from one generation to the next, because as we know, variation are most important for the process of evolution. So it conserves the chromosomal number of each species. It causes genetic variation due to the crossing over in the population of organism, and that leads to variation, and variation leads to evolution. Okay. And with this we have completed this chapter. So go through your NCERT line by line, also watching my video, and if you find you can share it with you. If you find it helpful, then you can share it with your friends. Like this video if you are new, and subscribe to my channel. Thank you. Jai Jagannath.